Okay, everybody. So real quick, the title of my presentation today is Canary in the Creek Bed, How Everyday Rain Becomes a Silent Killer in Our Streams. Let's get this started. So a little bit about me. My name's Nathan Ivy, and salmonids are really a passion for me. As a researcher, a fly fisherman, and a father, and protecting these iconic species isn't just about science, it's about ensuring that my daughter and future generations can experience the same thriving ecosystems that we cherish today. And I want to emphasize that this work is really collaborative, across universities, agencies, and community scientists. This is really much more than just a research network. It's a collective effort to understand how the waters we share are changing because they are. Now, first things first, let's briefly familiarize ourselves with some of the terminology I'll be using in today's presentation, starting off with salmonids. When I refer to them during this presentation, I'm generally talking about the anadromous species found in the Sound, and that's Chinook, chum, coho, sockeye, pink salmon, and coastal cutthroat are largely underrepresented salmonid with unique sensitivities. Um, these salmonids are uniquely adapted to their habitats and each with different challenges. And I want to emphasize that salmonids, especially anadromous ones, are very rare. Less than 0.5% of all fish species migrate between freshwater and saltwater, yet here in the Pacific Northwest, they dominate our rivers. These salmon hatch in streams, grow in the ocean, and return home to spawn. It's one of the most extraordinary life cycles and one of the most fragile. And so today we'll focus on the freshwater stages where stormwater pollution hits hardest. Now, um, salmonids are also a keystone species. And like the keystone in an arch, they hold ecosystems together. They bring marine nutrients into freshwater and terrestrial systems. So when they disappear, it signals broader ecological collapse and severs important connections and feedback loops. Now, I want to give you an example of what this looks like. Now, over the past decade, both Chinook and coho have gotten 10 to 15% smaller. This means less energy returning to rivers, about a 28% drop in marine nutrients feeding freshwater food webs. And for fisheries, that shrinkage has already cut value by more than 20%. Smaller fish mean weaker ecosystems. And when salmons shrink, entire ecosystems contract with them. Smaller fish lay fewer eggs, deliver fewer marine nutrients, and weaken those food webs from stream to sea. This means smaller salmon, smaller systems, smaller returns, and smaller futures. And salmon do a lot more than just live in ecosystems. They're actively building them. Every spawning season, they reverse the flow of nutrients from the ocean back to the forest, feeding trees, insects, and entire food webs. Their digging reshapes stream beds, oxygenates gravel, and creates habitat their young depend on. They are both um, they're both species and process, engineers of their own world. Salmon are seriously awesome. But wait, there's more. I want to briefly talk about sentinel species, sometimes called indicator species. And the use of canaries in coal mines is one of the earliest forms of biological risk assessment. They were effective because they reacted to carbon monoxide before any human symptom appeared. And ecological management today borrows this same idea, and sentinel species provide actionable early warnings. So, who plays that role in our watersheds? Coho are like the canary in the coal mine, only they live in the creek bed. Because they spend more than a year in fresh water, often in lowland streams, they're directly exposed to everything that washes off our roads. They're sensitive, protected under the ESA, and when they're distressed, it's the ecosystem's alarm bell. So salmon are extremely important because they're both keystone and sentinel. But how are salmon sounding the alarm? Urban runoff mortality syndrome, or ERMS, is a storm-triggered syndrome that kills adult coho before they can spawn. Symptoms include surface swimming, gasping, spiraling, all ending in rapid death. Um, Mortality rates regularly reach 60 to 90% in road-adjacent streams. And I want to reiterate, these are coho that made it home, but never spawned. Their eggs remain unlaid, their bodies unspent, and every storm now carries enough pollution to stop this cycle. Not over years, but within hours. So what's killing them isn't disease or drought, it's the water they're swimming through. Now, okay, have you ever seen a rainbow sheen on the road during a rainstorm or murky water gushing from a pipe into a creek? That's stormwater. As rain hits roads, rooftops, and parking lots, it's picking up everything, like I said. It's that oil, the tire dust, heavy metals, and it funnels it straight into our streams, untreated. We toxicologists call it urban soup. And according to the Washington Department of Ecology, each year polluted runoff transfers about 14 million pounds of pollutants into Puget Sound. Now, watch what happens beneath the surface during a storm. Um, Here you see an underwater time-lapse video of runoff entering Puget Sound. And you can begin to see particulates carried by that runoff. And I want to point out that what you can't see are the dissolved chemicals. And that's something that's really important to our work in the stormwater world. Ngone Blair, a PhD candidate in our labs, found that the dissolved chemicals in stormwater discharge can break down the blood-brain barrier in juvenile coho. This lets toxicants in, and the result is what we see in ERMS: disorientation, this erratic swimming, and the rapid death during storms. But what's really the culprit? Every time we brake, every time we turn or accelerate, tire particles are shed onto the road, the world's most overlooked pollutant. Rain mobilizes tire wear particles into streams, turning stormwater into a toxic delivery system. And tires are constantly being inundated with UV and ozone in the air, which cracks and ages rubber, as circled here. Now, to prevent that, manufacturers added 6-PPD. This is that chemical that reacts with ozone before the tire does. But that same reaction transforms 6-PPD into 6-PPD-quinone. Uh, you're going to hear that a lot this talk. And oftentimes it's the brownish sort of tire bloom that you see on the sidewalls of tires. It protects the tire, but washes off and poisons salmon at parts per trillion concentrations. Now, these concentrations we're talking about are almost unimaginable. I mean, they're impossible to imagine just about. And so to give you some context, one cooked grain of rice dissolved in an Olympic-sized swimming pool. That's all it takes to reach the lethal median, also known as the LC50. And at that concentration, half of the salmon die. And 6-PPD is essential for human safety. It prevents tire cracking and blowouts that save lives on roads. But as we look for a replacement, a PhD student in our lab, Anya McConaughey, works on this question. How will those alternatives affect salmon? Because whatever chemical comes next will still end up in our streams, and 6-PPD itself isn't disappearing anytime soon. That's why understanding synergistic effects is so important. This problem isn't simple, and it's not going away. I'm really grateful that we're working on this problem here at WSU. So, something that I think is really important in the story of stormwater and coho is the visibility, right? And visibility really matters. Adult coho are like really big fish. You can't miss them spiraling in a creek. Um, Juveniles, however, I've got these scaled, by the way. are camouflaged and very small and virtually invisible in flowing water. And if they're dying, we won't see it unless we're looking for it. So to find out what happens where no one's looking, we built a study inside a living creek. And Miller Creek was ideal. Spring storms consistently exceed 6-PPD quinone concentrations beyond lethal levels. That's that red line that you see. And this occurs just as young salmon begin swim up and out migration. So I want to talk about phase one of the study where we're testing real storms on young salmon. We started pretty simple. Two groups of young coho, one reared in clean well water and the other was exposed to live creek water during storms. And this setup let us see in real time how stormwater affects survival and behavior. Now, I want you to take a little bit of a tour with me. This is Miller Creek. It's a typical urban stream in our region, and I'm sure many of you have seen one just like it. And this video was taken just before one of our spring storms last year, and the creek, it's pretty calm, clear, and, seemingly harmless. And right here, you can see the intake basket here drawing unfiltered stream water directly into our field hatchery system. Notice that's 7 a.m. And here's a video I took at the same time inside the hatchery lab. In each aquarium on the right, fish are directly exposed to water from Miller Creek. This system allowed us to capture dynamic stormwater pulses as they occurred, which allows us to bridge that gap between static lab tests and full field studies. As the storm begins to ramp up, you can see this is just a picture, the video of the pump intake as things get going. And at peak storm conditions, the rising stormwater submerges that intake basket along with accelerated flow. And these tubes that you see there feed into an automatic water sampler, giving us a high resolution pollutant data throughout the storm. And this storm brought heavy sediment and an accompanying toxic surge. Now, these are conditions that were obviously mirrored in our raceways as evidenced here. It gives us a real-time look at how stormwater overwhelms juvenile salmon. But the real question is, what happened to the fish? And here's what we saw. The same syndrome that kills adults coho are also killing the juveniles. This juvenile coho is showing classic symptoms, not surprisingly, also triggered by stormwater. And in our Miller Creek study, naive juveniles exposed to real storms saw 80% mortality at each storm event. 80%. And this pollutograph shows how quickly toxicants enter streams during a storm. As stream flow rises, that's the blue wiggly line, chemicals concentrations spike rapidly. That's the red and black lines. And here in the yellow box, this is a 24-hour window where 6-PPD quinone levels exceed mortality thresholds. And here we can see this sort of sustained pulse of toxicity. And in Storm 3, that pulse led to 81.4% mortality, a single storm. So stormwater delivers really a double whammy to coho, and adults die before spawning and juveniles before migrating. And this pattern really scales up from individual fish to entire watersheds. Now, just humor me for a second here. I want you to imagine a migration of, say, 1,000 elk. And if 800 were struck by cars on a single stretch of road, it would probably make global news. We'd be demanding crossings, fences, investigations. Yet when the same thing happens to salmon, say 80% of 1,000 dying in a single storm, what then? Well, so far, the water clears, the creek runs silent, and no one even really notices. Because when death happens underwater, it's largely invisible. And when you can't see death, you can't measure it. That's why the Miller Creek Project is so valuable. It makes the invisible visible. And so this is what real-time and flow-through experiments can do. They capture stormwater as it happens in all its complexity, letting us observe mass mortality events in real time in non-adult life stages we might never see, and in pulses we can't even possibly predict. This approach gives us the ecological resolution to see the true impact of urban runoff. And urbanization amplifies every stressor. More roads mean more tire wear, more pavement means more runoff means more windows for mortality. And this transformation is incredibly visible in the lower Duwamish. Let me show you. So this is what the lower Duwamish once looked like. Notice this is a historic recreation based off of old maps. Essentially, the bend slowed water, um, and with these sort of wide meanders and floodplains and wetlands, these are like the wiggles that you're seeing. I want to point that out. This serves a really important purpose because it reduces erosion and creates a lot of refugia for salmon, especially young guys that don't swim very well yet. And another important thing is that these systems really spread nutrients and buffered those floods and supported an immense amount of biodiversity. But like we do, we straighten it out. So let's just take a quick look at what it's become. Now, not only was the Duwamish straightened, it was also severely deepened, replacing its bends with factories and docks. 97% of its original habitat is gone, and it became faster, hotter, and obviously much more deadly. Um, it's now become a highway for pollution, not a home for salmon. Now, this slide compares the historical meandering Duwamish to the highly channelized version we see today. And channelization increases flow velocity, reducing refuge areas for salmon and forcing them to expend more energy during migration. According to a recent study by Vels et al., avoidance of high velocity gradients results in delays up to eightfold, with fish traveling 3.5 times further to avoid these conditions. This dramatically increases energy use, making some salmon much more susceptible to stress, illness, and mortality before they can even reach spawning grounds. Now, the same efficiency that benefits us humans breaks salmon. I want you to take a moment and look. Can you spot any other differences? Now, in natural systems, rain soaks into soil, replenishing groundwater and cooling systems. But when we pave that land, the rain has nowhere to go. It flashes off roads and rooftops, extremely fast, hot, and dirty. Pardon me. This really overwhelms the streams and essentially suffocates salmon habitat. So, like I said, like in natural systems, rain infiltrates the ground. That's really important for replenishing groundwater and maintaining regular stream flow. But in urban areas, impervious surfaces like roads and parking lots block that infiltration. And this causes a dramatic increase in that surface runoff that I talked about. This increased runoff carries pollutants, erodes streams, and reduces the groundwater recharge. So essentially, it's disrupting the flow that sustains healthy ecosystems. And urbanization shifts the water cycle entirely, creating conditions that amplify stormwater impacts. Now, the road effect zone illustrates how infrastructure fragments ecosystems on a massive scale. Roads, which occupy just 1% of land, disrupts 20% of ecosystems by severing migration pathways and altering those stream flows while also being a vector for introducing pollutants. And for Washington's salmonids, these disruptions are compounded by over 20,000 culverts statewide, blocking access to more than 5,000 miles of habitat, but physical fragmentation is only part of the story. Urban runoff introduces chemical barriers like 6-PPD quinone, PCBs, and PAHs, transforming reconnected habitat into lethal ecological traps. So, addressing these dual barriers, fragmentation and toxicity, is really critical to ensuring the survival of species like coho, chinook, and steelhead. Now, um, culverts have been a major topic of discussion in recent history, and perhaps you've probably even experienced traffic as a result of major culvert retrofits occurring in Washington State. Now, the current WDFW strategy uses a scoring system to prioritize culvert removal and replacement. This is generally based on habitat length, ESA status, and cost, but it doesn't account for urban gradients or stormwater toxicity. That means millions, over $1 million a day are being spent to remove these barriers without checking to see if the water behind them is survivable. And here's a fish passage project with a newly open stream. But if you look closely here at this red arrow, that pipe is still discharging untreated runoff. So while we removed the gate, we haven't done anything to prevent this ecological trapping. So what do we need to know about culverts if the goal is to restore a migratory corridor? The Seattle Times created a really compelling article that visualizes the challenges that salmon face on their migratory journey back to spawning grounds. Now, as we watch this clip, let's examine not just the physical barriers, but how each of these structures becomes a vector for toxic runoff, ultimately compounding the threats to salmon survival. So first, there's, let's see, velocity barriers. Now, these are narrow culverts that increase stream flow. They erode sediment and release roadway pollutants like heavy metals and PAHs into the water. Next, there's these slope barriers, which are really steep culverts that speed up runoff, transporting contaminants downstream rapidly and further degrading habitat quality. There's trash grates. Those are designed to trap debris, obviously. They also accumulate organic matter, which decompose and release pollutants into the stream. There's depth barriers, which are shallow water and culverts with reduced flow, which really concentrates toxics like 6-PPD quinone and PCBs in stagnant areas. Then there's the headroom barriers, where sediment buildup constricts flow and traps pollutants, making it really hard for salmon to pass through and survive. And then finally, there's probably the most famous, which are outlet drop barriers, where high drops create plunge pools, where stormwater toxics get resuspended, exposing fish to increased contaminant loads. I'm sure many of you have seen videos of fish trying to jump into these outlet drop barriers. Now, each of these barriers really represents a missed opportunity. Without addressing these sources of pollution alongside culvert removal, we risk restoring access to habitats that are toxic to salmonid life. And this salmon stress meter here shows how water temperature affects salmon survival. At optimal nursery temperatures, 8 to 12 C, growth and development really thrive. And as temperature rises, stress increases with significant mortality risk above 18 C and survival is compromised beyond 22. Now, this gradient is really critical for understanding the impacts of warming waters, as we'll see in the next few slides. So I performed a little GIS analysis using the Norwest data set from the U.S. Forest Service, which provides a detailed weekly maximum temperature data for streams. This represents the highest 7-day rolling average of daily maximum water temperatures. This is a very important metric for assessing salmon habitat suitability. This data set was chosen because it captures acute thermal stress conditions, which is really important for helping us understand how heat waves, urbanization, and stormwater runoff create thermal barriers or mortality risk for salmonids. Now, the following slides will show how suitability has changed over time, illustrating key water temperature trends for salmon resilience. Okay, so back in 1993, which was the beginning that this data set um, had access to, salmon suitable waters were really dominated by ideal nursery conditions. And I want to point out that everything here is color coded. Took a lot of time to put this together, and you can see where all the mountains are. Further away from cities, you have these really nice dark blue colors. Um, and so over 34% of streams had these optimal temperatures of 8 to 12 C, while stress-induced waters above 18 C were pretty rare, making up only 14% of the total. And the histogram on the right there, if you look closely, you can see how um, there's a clear dominance of cooler water bands. These conditions really supported healthy salmon populations, but over the next couple of slides, we'll see that this balance has really shifted over the past few years. So fast forward to today, and we see a very drastic loss of cool nursery waters, down from 34% to just 19. And at that same time, stress-inducing and lethal waters have really surged. So streams with temperatures above 22 C, which were once extremely negligible, now make up over 10%. That's a tenfold increase. So the histogram highlights really this growing dominance of warmer bands, which pose severe risk to salmon survival. And by 2028, in this projected model, salmon suitable waters are in significant crisis. Optimal temperatures are projected to drop below 10%, while the hottest, excuse me, most lethal waters surpass 16%. The histogram really shows that this is a very worse, it's a worsening trend, right? With stress and lethal waters at this point now dominating. Salmon face these shrinking habitats and rising thermal barriers, which severely impact their survival. And here's just like a picture of that kind of transition. Okay, so overall changes. Here's really the big picture. Over 3 decades, we've seen a dramatic shift in salmon habitat suitability. Optimal nursery waters have dropped by 74%, drastically limiting critical rearing areas. These are nursery waters we're talking about. Meanwhile, stress-inducing waters have increased by 170%, pushing salmon beyond their physiological limits. Most alarming, lethal waters have now surged by 1,500%, creating deadly conditions in once thriving habitats. Now, this trend really shows us that there's an urgent need for mitigation to protect salmon from a future where suitable waters are nearly gone. And I think it's really important to understand that salmon are cold water specialists. They require cold water in order to thrive. So I'm going to belabor this a little bit. Warmer waters force salmon to burn energy faster, leaving them more vulnerable to stress. And these conditions also amplify bacterial and fungal infections, which further weakens their resilience. And as salmon migrate, the combination of exhaustion and infection often results in death before reaching spawning grounds. And for those that survive the journey, the stress reduces their capacity to tolerate toxicants, compounding the impacts of polluted waterways. Now, this cycle really demonstrates that there's this intersection that we need to be paying attention to of thermal and chemical stress on salmon survival. So I ran a pilot study last year and this year. Essentially, let me give a little background here. So stormwater in urban areas carry these superheated waters from impervious surfaces. It creates these sudden and really extreme thermal spikes in streams. And these rapid changes, known as thermal shock, essentially occur when water temperatures increase by 4 C or more in under 24 hours. This really overwhelms salmon physiology because they're cold water specialists. And in this pilot study, we exposed juvenile coho to 6-PPD quinone under both acclimated and thermally shocked conditions. Mortality increased by 132.56% in fish subjected to thermal shock. What this does is it shows us how flashiness really amplifies toxic effects. And the pilot um, shows us these really lethal synergies that occur between urban heat islands, stormwater flows, and chemical contaminants. So, in short, thermal stress layers onto chemical stress, pushing salmon beyond their physiological breaking point. So, um, when I emphasize that thermal shock isn't the only challenge, right? And I want to do a little bit of a recap. So, channelization, this is what kind of confines flow, right? It increases velocity most often and prevents refugia in side channels, for example. Um, and then there's rising temperature driven by urban heat islands. Now, these are really important to understand because this reduces oxygen and push salmon beyond those thermal tolerances. Culverts create physical and chemical bottlenecks, which really concentrates contaminants and simultaneously delays migration. And impervious surfaces, like we've talked about, they accelerate runoff, delivering these pulses of stormwater directly into streams. So together, these amplifiers compound stormwater toxicity. This really creates a sort of lethal cascade for salmon in these urbanized watersheds. Okay, so I want to talk about phase 2 of the study, where we looked at stormwater's effects on the entire early life cycle, from embryo to juvenile. And really the goal was to see what chronic exposure does over time, not just during storms. So let's start with embryos, how the early exposure begins. So a little background. Embryos are protected by the chorion, which is what I've got highlighted here. It's this outer membrane. That barrier may buffer early exposure to stormwater, but it's not impermeable. And timing really matters for these things. So to test that, we exposed a lot of embryos to live stormwater from fertilization onward. And we want to see what happens. And so I've got some images and some videos that I want to share with you so you can kind of see what I've been working with. Now, here's a picture of a dechlorinated embryo. They're incredibly beautiful creatures. One of my favorite parts of working in this field is the opportunity to really look and observe and behold these fish. And from October to April, last October to April, we ran the largest ever chronic creek water study using this same real-time system where we captured every storm rather than just one at a time. And what we saw in those early stages showed a subtle but very serious impact. And so what we did was we examined mortality trends, RNA expression. We are still actively measuring tissue chemistry and morphometrics, which is just imaging really scientifically and then measuring things individually. And so what we're doing now is we're learning how this chronic exposure to urban streams impact these early life stages. Now, here in this picture, you can see major inflammation occurring at the gills, around the eyes, and then you also have an underdeveloped sort of lower jaw. Now, I want to give you another example. So in the embryo phase, we observed localized blood pooling near the retractor lentis. This is a muscle that controls lens movement and focus. So this was only seen in the chronic stream exposure group, and it became more frequent near hatching. So this capillary level pooling suggests that there's an increased vascular permeability in these chronically exposed fish. And this really matters because vision drives foraging, predator evasion, and migration later in life. So damage here, while in the embryo phase, impacts downstream survival and behavior. In ecology, we call that sensitive dependence on initial conditions. Now, fast forward to the elven life history stage once they've hatched, um, and they still have their yolk sac. So some of the things that we saw. Um, so let's take a look here. Here we're looking at coho at the same developmental ATU, but as you can see, they're not developing at the same rate as their well-grouped counterparts. Stormwater exposed fish show delayed yolk sac absorption, which creates more than just a timing issue. It can lead to delayed readiness, which is so that's slower growth. That means that they'll swim up later, and that gives them a much, so that leads to a competitive disadvantage of sorts. Now, in the elven life history stage, we saw 13% mortality during the first storm post-hatch, at a very small concentration of 6-PPD quinone, but mortality dropped in subsequent events, which raises a really important question for us toxicologists. Can sublethal stress prime survival? And if so, what are the long-term costs of that plasticity? Now, I want to point out that this is probably the first ever footage of this urban runoff mortality syndrome impacting elven in an environmentally relevant context. Um, and I've got the same for fry as well here. So this fry, for example, has already absorbed its yolk sac and has begun actively swimming. So whenever I say swim up, that simply means that once they've absorbed their yolk sac entirely, they swim up to the surface, take their first sip of air, which inflates their swim bladder, and then they begin actively swimming, as well as exogenously feeding. That just means feeding on food outside of their yolk sac. So ERMS still occurs at this stage, but mortality is lower than in the elven life history stage. And this shift may indicate an adaptive threshold shaped by those earlier low dose exposures. And these are representative fry from our control and exposure groups. And I want you to notice the tail fin. Chronically exposed fry have visibly smaller fins with distinct shapes, which are wedge versus fan, wedge versus fan. Now, here, I want you to kind of look with me as we kind of compare and contrast these images. I want to point out there in the center, you can see side by side of caudal fins. That's a tail fin. Their lengths are very different. And the operculum, which are the gill plates there, if you see the fish on the left in the creek exposure, you see a lot of them are smaller. They're exposed, exposing their gills. And then, of course, there's quite a few craniofacial effects too. And here's a little bit of a close-up of that tail fin difference side by side. Now, the exposed fish had that narrower fin with reduced surface area and stiffer margins. And this really limits what's called lateral flexion and thrust during swimming. So this shrinks both propulsion and maneuverability. And it's a subtle change, but with very big consequences for survival. Now, to quantify these fin differences, we measured the fin surface area and normalized it to fork length. That's where we saw that 30% reduction in the surface area. This isn't just a structural shift, right? Because fins drive swimming, and a smaller fin, that can mean slower escape, much weaker migration, and reduced fitness downstream. So to test their performance, I ran a series of swim tunnel trials with the fish of both with the fan shape versus the wedge shape. And in these stormwater exposed juveniles, right, they recovered more slowly and failed more often. And so This reduced endurance also means fewer escapes from predators and less energy left, and less energy for migration. So even when fish survive exposure, they're oftentimes emerging much weaker, and less ready for the challenges of the world. So their environmental cues, they're not able to respond to them as well. So real quick, I also did a very cool study this past summer. And, so coho, they really gave us the first clear signal of stormwater toxicity, right? But they don't live alone in these creeks. Coastal cutthroat share the same water, the same storms, and the same stress. In this year's comparative spring study, spring/summer, both coho and cutthroat exhibited ERMS-based mortality, yet cutthroat were even more susceptible. And my ongoing genome-wide association study will let us know whether there's some genetic differences in detoxification or stress responses, and hopefully that can explain the vulnerability as to why some die and why some survive. We're still trying to figure out so much about this subject. So in all, Miller Creek is a rare site where real-time methods showcase both lethal and sublethal stormwater effects. Mortality isn't restricted to just adults, and chronic exposure disrupts development and alters readiness, with early life stages being especially vulnerable. So what's next? This fall we're doing a side-by-side comparison exposing coho and steelhead or rainbow trout, Oncorhynchus micus, to the same embryo to juvenile life history stages. Very exciting. Please stay tuned for all of that. But I want to point out that it's not all bad news. I know that I've sort of been casting this as a major crisis, which it is, but there is some hope. Some solutions are within our reach, and they're already proving to be highly effective. And so green stormwater infrastructure offers it's a proven solution to urban runoff toxicity. Bioswales, rain gardens, and detention basins can effectively reduce salmon mortality by up to 90% because filtering pollutants like 6-PPD quinone really matters for fish. But despite its potential, GSI is entirely absent from the WDFW strategy, which really leaves urban watersheds very vulnerable. So aligning these GSI retrofits with culvert removal projects is really essential to ensuring that reconnected habitats are viable for salmon recovery, especially in urbanized areas. These videos are from an experiment where fish were exposed for 24 hours to either clean water runoff or runoff filtered through bioretention. And at 4 hours, the fish in runoff were already sick and died soon after. But that same water passed through bioretention caused no acute effects. Shown here for adults, but we've also shown this for juveniles and alevin. Now, although we're really proud of some of these efforts that we've made to green our cities in the Puget Sound Basin of Washington State, there's still a lot of impervious area to compensate for. Excuse me. Best chances are really in new development where GSI is mandated. Now, the salmon was our canary, dying first from 6-PPD quinone in stormwater. And now we're finding the same compound in human urine and liver cells, which essentially triggers oxidative stress and disrupts metabolism. A study recently done last year demonstrates this. And so the warning is really clear that what poisons the salmon is beginning to reach us too. And in closing, I want to leave with you a quote. The Pacific Northwest is simply this: wherever the salmon can get to. This is not a metaphor. It's a boundary line drawn not by politics or property, but by possibility. And where salmon run, life flourishes. And where they can't, we lose the living bridge between ocean, forest, and people. And with that, I'm now willing to answer any questions that you may have, and I'll stop sharing.
Thank you so much, Nate. Big round of applause. That was an amazing talk. I'm sure we all enjoyed it and learned so much. So I do have a couple questions in the chat, so I'll read those to you. And if anybody else has questions and feel comfortable, you can raise your hand or you can just keep putting it in the chat. So question one was for phase one by, this is from Ariana Wald. She says, have there been any studies to compare 6-PPD-Q levels in a stream where a heavily vegetated riparian area versus a sparsely vegetated stream?
I love this question. There's not any studies that I'm aware of where they've been monitoring this, but there is active research right now looking at how plants could be used to potentially mitigate the overall effects of 6-PPD quinone and their levels. So I think that's a great idea. The stream that I'm working in has kind of a mixed um, it's a little bit of a mixed bag, but yeah, great question. There's not any studies that I'm aware of.
Yeah, and related to that from the same person, Ariana, is regarding green infrastructure, does 6-PPD-Q settle out of stormwater or is it taken up by plants?
Ah, another good question. Um, that's still actively being studied. So I want to point out that 6-PPD quinone is a relatively recently discovered chemical. And and so we're talking 2021 was whenever the main paper was published. Our lab here at WSU was the lab to identify it. And so a lot has happened since that discovery, but we still don't know um, what its fate in transport really is. Uh, the really complex and tricky thing about urban streams is that there's so much that's going on there. So many, so much sediment, so much debris, everything kind of, it's a mixed bag in that urban soup whenever it comes downstream. So trying to figure out what plants are good at like bioaccumulating that is something that's an active research question. And people at our lab in the Green Stormwater Infrastructure group are studying that. But it's still a major work in progress. Great question.
Yeah, I was just going to note that there is a PhD student, Nandini Premkumar. She's studying with Ani Jakaran, and she is looking at 6-PPD-Q uptake in a few crops like barley and wheat, and seeing how they're kind of how the transport, I guess. All right, and then one more question from Ariana. Thank you so much for all the questions. Is there any thought of studies exposing hatchery salmonids to varying levels of 6-PPD-Q and comparing their development and mortality rates?
Ah, I am so glad you asked this question. This is essentially what I did with our Miller Creek study. So we took embryos from hatchery settings, and we have essentially wild and hatchery proxies. And so one of the tricky things about working with wild versus hatchery stocks and trying to understand their impacts is that hatchery salmon have a very different life experience, and they're generally held well, not all of them, but in general, they're held at a steady state, a steady temperature, which temperature really drives their productivity and their growth rates. And so oftentimes you see hatchery fish enter into the stream earlier um, than their wild counterparts and at 2x their size. And so they're really competing with their wild counterparts and essentially displacing them. But what we've seen in the Miller Creek studies is that these hatchery fish, which are essentially naive, unexperienced fish, when they are exposed to stormwater, they have a very acute responses, much different than the acclimated or chronically exposed fish, which would be considered your wild proxies. So yes, I am actively studying that. I have partnered with the Wild Fish Conservancy, where we are also studying that, and we're looking at the impacts of, well, because essentially all of the research done to this point in lab settings has been done exclusively with hatchery fish. And so and they've been held at conditions that mirror those of hatcheries. So we've essentially been making a lot of decisions for hatchery fish rather than the wild fish that we're trying to protect. And so the Miller Creek study provides a really unique look at what essentially the urban gradient life history is for them. So lots of really cool research happening in that domain with hatchery versus wild. Great question. Thank you for asking that.
Any other brave souls want to raise their hand and ask a question? Or if you're shy, you can type it in. I have a question, Nate.
Sure.
What do you want to do after your PhD?
Uh, Continue to work with salmon. I'm very interested in dam removal, culvert removals, and so forth. I really want to see salmon back to historic natural abundance at one point, but who knows how that'll go.
Yeah. Okay, one more thing I just thought about is when you're showing how WDFW is doing all those culvert removals, but they're just piping straight stormwater into the creek, why can't they just install a little, I don't know, rain garden or something like, bio.
Yeah, so that really comes down to a lot of times these are piecemeal type of operations where you have some that are being funded by different groups and some of them have larger budgets, but it's not something that they've taken into their draft strategy. And so I did go in and speak at these public sessions whenever they were presenting this draft strategy and pointed that out. And I have since had quite a bit of contact with the WDFW project managers about this subject because one of the things that is concerning and the entire reason why stormwater research has begun with salmon in the first place is this notion of ecological traps that I briefly touched on. Some context so that everyone knows a couple decades ago, salmon were in major decline here in our region, and we decided to invest, simply because, hey, like we're Seattle, like we're supposed to have like salmon. We're a salmon nation, right? Salmon people. So we should kind of have salmon in our streams. And so we spent millions of dollars trying to restore creeks, and one of them was Longfellow Creek. And so we removed culverts. We did all sorts of riparian restoration, and sure enough, when we did that, salmon came back and established that section of the creek. And while champagne glasses were tinkling and cigars were being lit in celebration of salmon coming back, one thing that occurred was a rainstorm, and every single coho in that creek died. Every single one of them, 100%. And that obviously sent us down this huge rabbit hole for the next 2 decades where we've been studying this, what in the world has been happening. And we found that, first of all, those fish were dying before they even spawned, which, this is something that's been happening for a long time. But this whole notion of an ecological trap is what Longfellow Creek represented. And so whenever restoration occurs, we need to make sure that we're also checking back and doing long-term monitoring to make sure that the waters are going to be safe for salmon once they do come back. So it's been a very interesting ride. And I am working with King County actively on the subject of culvert replacement as well as stormwater mitigation. Okay. Let's see. I see another question here. Can you explain the 6-PPD quinone leaching from stockpiles of tires exposed to ozone, more or less than tires on cars? So, Krista, I'm assuming that you're talking about like whether it's coming from like the tire reefs that are in or recycled ones, so like on AstroTurf fields or something like that. Um, so That's like a major source of research right now. Like a lot of people are asking that question. I don't interface with that directly. This year, next year, we will be sampling and exposing fish to effluent from an AstroTurf football field on Miller Creek, and we'll be looking at that specifically and whether or not these fields are um, producing 6-PPD quinone at rates that are lethal to salmonids, because obviously um, those fields are everywhere now. And then in terms of like the tire reefs that are in estuaries and in streams, that's something that our collaborators over at University of Washington are actively studying, where they're looking at the life history of these tire reefs through their wet dry cycles, which is something that's pretty fascinating. We're still waiting for some results on that. Great question.
Nate, do you mind if I share your email address with everyone in the chat?
Yeah, please do.
Okay, I had a request for that, so.
And if you have any questions, please feel free to reach out. I love getting emails, and I'll do my best to try and answer them. This is something that I care a lot about, if that wasn't already demonstrated. And I want everyone to know uh, uh, everything that I know as it comes through. So I'll continue to keep presenting as this research gets updated. Um, and Stephanie at the Washington Stormwater Center is also doing great work communicating a lot of the work that our lab's doing, which is much more than what I'm doing.